So good afternoon, everyone. My name is Adam Knoll. I'm the Vice Chair of the IEEE Communication Society Technical Committee on Molecular, Biological, and Multiscale Communications. I'm very pleased to welcome you to our inaugural session of our Molecular Communication Outside the Box, Breaking Disciplinary Boundaries. Yes, so this is a new session that's being run by our newly appointed chair here for our by the committee. Let me just double check the title here. Yeah, so we have the technical committee's educational services coordinator, which Max Schaefer has gratefully agreed to take on. So part of this role will involve providing and organizing activities for members of our community, both to learn and make the field more accessible to people that are in potentially tangential fields. And so there'll be kind of two elements. One will be activities that are, let's say, tutorial in nature, that are intended, let's say, in-house, so more within the field as we know this and it has been established. And then the other track will be along the direction of what today's session will be, which will be bringing in people that really come from outside the molecular communication space, but who we believe have great complementary expertise and that we can learn from as this interdisciplinary field expands and grows. So I'm very happy to hand it over now to Max to introduce our first speaker. And I hope you find these sessions useful. If you do have suggestions for types of talks you'd like to see, coverage you'd like to have covered in future sessions, I'm going to take this opportunity to invite you all to flood Max's email inbox with suggestions. He's giving the thumbs up, so we're welcome to do that. But yeah, I'll pass it over now to Max to introduce our speaker.
Sure, thank you, Adam, for the nice introduction of this series of talks, and I'm very happy that this is happening now, and I hope also we will have very good more talks in the future. So, but now today we have the honor that Friedrich Simmel will start. So he already gave a very nice talk on the molecular communications workshop in Erlangen this year. And then I also heard him on another workshop in Munich where we have attended together. So he's really doing amazing work. So maybe a bit about Fritz. So he's done his PhD at the LMU in Munich. And after that, he was a postdoc at Bell Labs, where he starts with DNA nanodevices. Then after having joined a research group, he's now since 2007 at the Technical University Munich, so same city, but different university, and holds the chair of physics of synthetic biological systems. He is also was a coordinator of a German excellence cluster, and he's a member of the German Academy of Science and Technology, was awarded with ERC grants, and is also a Max Planck Fellow life member. So yes, so there are a lot of more things to say, but I think that is enough. So the stage is yours, and we are really curious to hear about genetic switches and circuits based on nucleic acids.
Okay, great. Thanks, Max, for this really very nice introduction, and also Adam, and thank you for inviting me to this inaugural session of your seminar series. I was not exactly sure what to prepare and for exactly which audience, so I might completely miss what you are expecting, or maybe some of the things you might find interesting. I try to explain a few things which might be too basic for you, and maybe others will be maybe difficult to access. We will see how this goes. So one thing is that actually I have a physics background, and I was always interested in also information processing in some way, but we happen to be in a biophysics environment, bio-nanotechnology. So there is always the question, how could we actually implement computational functions in biological systems or biomolecular systems? And I guess also for synthetic biology, electrical engineering and computer science was kind of a role model, so that obviously computer technology is an extremely successful technology that has been scaled up over many, many orders of magnitude in terms of computational power and all these things. And I think that many people would dream of making something similar with biology. And I guess one important point for the development of synthetic biology was that the idea is maybe if you could sort of modularize somehow biological systems enough and describe their functions in some type of high-level programming language type of way, maybe you could actually build up more and more complicated systems. And in the end, unfortunately, biology is kind of a very different technology than kind of, let's say, silicon technology or silicon-based chip technology. And many of the things are completely different, and some things don't work at all. And it's very difficult to get some of the experiments running, at least at the moment. So therefore, these very high ambitions are probably not yet met. But I guess it's useful to think about kind of trying to sort of implement that and also see what are the differences. Because also it's interesting to look at how differently is actually our biological systems processing information compared to kind of human-made systems. So I guess it's also useful to learn from the differences. And so here is a slide. I'm putting up that slide because I usually forget that at the end. I wanted to thank my group and the funding agencies. And what I'm going to talk about today is mainly actually due to only 3 people. The 2 just already left the lab, mainly working on these RNA-based switches, and Tom was working a lot on kind of random RNA and DNA sequences, which will become clear later in the talk. So I think that what I just said is somehow recognizing that some of the things or some of the let's say properties that we would expect from, for example, robotic systems are actually also realized in biological and biomolecular systems. So for robots, we usually would integrate, for example, sensors, computation, some computation that decides actually which action to take based on sensing. And there are other kind of aspects, of course, involved, how to supply like a robotic system with energy, how to communicate between subsystems, for example. And then, of course, like a big question is how to actually implement learning, how to sort of let a robot, maybe how to train a robot to perform certain actions better or not. And if you look at that, actually somehow also with biomolecules or cellular systems, you can also implement these functions, or they are already implemented in a sense. So what I show on that slide is that's just a single, it's just a molecule. This happens to be one of the bases that you need in DNA. So I guess it's guanine, and here is some RNA molecule, which I'm going to talk more about later. That's kind of a so-called riboswitch. That's an RNA molecule that can change its conformation. This is a bacteriophage, like a little virus that infects bacteria. That's an E. coli bacterium, or it's supposed to be one, and that's a eukaryotic cell. And you see kind of all kinds of structure in there. They are on very different length scales. So this is molecules only about a nanometer in size. Something like that would be several nanometers. That's probably on the order of 100 nanometer. That's about 2 micrometer in length. And here you might have something between 10 to 100 micrometers. So that's actually very different length scales. But, and I guess there's a relation between like how complex these systems is and what you can probably implement with them. So I guess that, for example, sensing usually would mean that some signal, some input makes a change that you can detect. That's kind of simple. So this could be actually realized in molecular switch. For example, a molecule binds to such an RNA molecule, it changes its shape, and this can be seen as a as a sensing event, and this is actually also used in cells in certain contexts, for example, in ribo switches, which I'm going to talk about later. But of course, there are also other sensor elements, for example, surface type of elements on cells that have all kinds of receptors. But then there are also elements that allow actuation. For example, there is a flagellum here that allows the bacteria to propel inside of such viruses. Sometimes there are molecular motors that help to push DNA into the capsid or into the cell, depending on how you want it. Inside of cells, of course, you have molecular motors that shuttle things around. I'm not going through the whole list, but in principle, you can find like all these elements that we would also probably find in a robot, but they are just made with a completely different technology. So one thing that I need for the rest of the talk, which you probably all know from your biology classes, or maybe you are already working with biological systems, so that's kind of trivial for you. What's actually going on inside of these cells? So here's the cell nucleus of a eukaryotic cell that contains some DNA, and that is the nucleoid or the chromosome of a bacterium. So this is DNA, and what cells do with DNA is they translate the DNA into RNA, and there's a protein that is responsible for that, that's called RNA polymerase. And usually an RNA polymerase recognizes a certain sequence on a piece of DNA, and that is called the promoter. So the RNA binds here, then RNA polymerase binds, it makes RNA, and this RNA molecule comes out of the polymerase, and then another big kind of molecular complex comes in that's called the ribosome. It assembles on the RNA, and then it makes a chain of amino acids, or it connects amino acids into a chain in a process called translation. And this amino acid chain, if it's long enough, it might fold into something that we then call a protein. And this general kind of idea that there is some information stored in the cells in the form of DNA, which is then transduced to a form of RNA and then protein. That's the famous central dogma of molecular biology that was actually formulated by Francis Crick already quite some time ago. So one of the people who actually kind of elucidated the structure of DNA later came up with that central dogma. So you see that the information flow is supposed to be from DNA to RNA to protein. What you also see is that in biology, you also have other possibilities. So one thing is that for a cell to replicate, it also needs to replicate its information. That's this kind of loop where DNA makes more DNA. But then there are also other processes where, for example, RNAs is translated back to DNA in a process called reverse transcription, which you find in RNA viruses, for example. The coronavirus needs to sort of make use of that process because it's an RNA virus. To come to the topic, what I'm actually want to talk about is actually making synthetic systems from mainly RNA and sometimes DNA to perform simple computations in cells that may be used to program cellular behavior, to maybe add or manipulate functions that are already there. And for this, I briefly want to talk about how DNA looks like in the first place, and then RNA. So also this is textbook kind of knowledge. So DNA is a double-stranded molecule. A double-stranded structure, which is formed by two DNA strands. And with that, I mean a single strand of DNA is just like a polymer that is made from subunits. And these subunits are called nucleotides. And each nucleotide is one of these little sugar molecules. That's a so-called deoxyribose. So it's a 5-carbon sugar And each of these sugars is connected to one of these molecules, which are called bases. And here you see the 4 bases in DNA. That's the famous 4 letters: adenine, thymine, guanine, and cytosine. And so these little units here, they are all connected to each other via these these phosphodiester backbones. So these actually all carry a negative charge, and therefore the whole molecule is quite negatively charged, which is kind of important in many contexts. And in order to let, like if 2 of such strands happen to be complementary, then they can actually bind to each other. And complementarity means that a guanine base can actually bind to a cytosine base, and an adenine base can bind to a thymine base. So they can, if one of the sequences in one strand matches the sequence in the other strand, they can bind to each other, and then they will make actually such a double helical molecule. And that's the famous double helix. If the 2 sequences don't match each other, so if there's, for example, a C on the other side, instead of the A, then C and T will not base pair, and then the whole structure will be energetically less favorable. And if there are too many of these mismatches, then the 2 strands will just not bind to each other. And this is something that is regularly used in all of biochemistry and molecular biology, and also in biophysics, what I'm going to talk a little about. Then there is also this other molecule, RNA. So here, that would be kind of what I have drawn here. So this double helix really as a molecule looks more like this here as a molecular model. And this molecule here, RNA, would be actually similar to DNA, but it's a single strand. And here you have a slightly different sugar that's called ribose. It has this OH group instead of only a hydrogen for deoxyribose. So here you see the difference between these 2. And it also has 4 bases, but instead of thymine, it has uracil. So in RNA sequences, you always see a U instead of a T, just as an explanation. So in a cell, you typically find DNA in double-stranded form, and in contrast to that, RNA is usually single-stranded, and therefore with the RNA single-strand, you can do more things because the single strand can bind back to itself. It can make kind of interesting structures, and this is what we are going to talk about later. I have just this little slide here just to say that a part of my group is working mainly on DNA nanotechnology, which is not really related to the topic of the talk, but I just want to say that people actually use DNA molecules to build all kinds of crazy kind of structures. And this is based essentially on that double-stranded molecule, but you can actually connect several of these double strands together by, it's like making a fabric from DNA. And this is shown here. So here you see, for example, a plate that consists of many of such DNA double helices that are all kind of connected to each other. And you can really make like little mechanical devices of that. So this would be some like long kind of arm consisting of 6 double helices that can rotate on that plate. Or here would be a different structure where you have a ring that is can move on an axle made from DNA. So here you see a molecular model of that, and these are actually electron microscopy images of these structures. And so I guess this is actually quite an amazing achievement in this field, that with the so-called DNA origami technique, we can really use essentially computer-aided design programs to make structures like that. You can make the molecule, take an image of them, and they look more or less the same. So that's I think quite impressive. And so this is not, of course, not our achievement. It's kind of the result of a decade-long kind of efforts by many people in this field. And it's, in my opinion, one of the first sort of technologies that really allow you to sort of describe molecular components and functions on a relatively abstract level, where you have computer-aided design programs that can be then sort of translated into real-world molecules that have some function. And maybe this will be true for all kinds of other like real biological functions in the future. So what I wanted to talk about today is kind of dynamic DNA and RNA devices for gene regulation. And one of the points with the DNA double helix is that it's more or less inert. So maybe going back here, so if the two DNA strands are bound to each other, then they are bound to each other, and you can only rest them apart by, for example, heating them up. So if you heat a solution with double-stranded DNA to 80, 90 degrees, then the two strands will fall apart. And then you might use them to do something else. Then they could bind to another molecule. But typically, this is not what you can do in a cell. So cells have actually enzymes to separate the two strands from each other, which is important, for example, for DNA replication. But here's a trick how you can also achieve that. And this is something that is frequently used in the rest of the talk. So for example, this is a schematic depiction of a double strand where one end usually carries kind of a little arrow. This is because DNA and RNA have a direction. So here you see this is called the 5' end, this is called the 3' end, and the 3' end gets that little arrow, and the two strands bind in an anti-parallel fashion. So usually your structures always need to be made such that the two strands show in opposite directions, and this is what you see here. So there's an arrow here and an arrow here. That's where this comes from. And if only a sequence A would be bound to its complementary sequence A bar, then this would be just a duplex, which would be relatively stable. But you can actually use a trick to somehow remove this sequence A bar from the other sequence in a reaction called strand displacement. And for this, you add a little sequence tag here called toehold. And if you now mix this structure with another molecule that looks like this here, then this little piece B will bind here. It will make such a structure, and then the red sequence actually is exactly the same sequence as the black sequence here. So what then will happen is that simply because of thermal fluctuations, for example, this base pair would break open, and then this base can bind here, and then maybe the next breaks open, and this can bind here, and this will go back and forth until at one point this black strand has been displaced. So this is why it's called strand displacement. And because the blue sequence in the end, or this complex here is more stable than the initial complex, this is actually thermodynamically favorable. And you might wonder why you want to do this. So the point is that you can actually come up with more complicated situations where this allows you to switch a DNA or RNA molecule from one conformation to another that has some meaning later on. And I think that this will become clear in the next slides. So one example is shown here. Assume that you have something hidden in such a hairpin. That means that this sequence B is not really accessible to another strand because the strand cannot wind through this kind of hole here because it's too bulky. But if you, for example, add a DNA strand to this structure, then this C bar acts as a toehold. It will bind here. It will break this double-stranded kind of stem that will create such a structure, and then the sequence B that is formerly hidden in the loop will suddenly be accessible. Then another DNA strand could come in, or RNA strand, and bind to this sequence. And here you see that in principle, the input, this red sequence, which you can treat as an input, activates the availability of this B sequence. So this is a very simple form of kind of switching, if you want. And you can also do this in a more complicated fashion. So people actually created systems like this that can actually also make chain reactions where you make very long or big structures, which has actually already found use in biosensor applications, which I'm not going to talk about now. So, so coming back to the initial kind of motivation, you can now actually use such tricks with RNA molecules inside of cells. And the point is that actually nature already uses something kind of similar. In bacteria, you sometimes find RNA molecules that have certain structure in the molecules, and so now I will jump a little back to my introduction here. So this would now refer to one of these RNA molecules. So the in a bacterial cell, for example, an RNA polymerase has generated this RNA molecule, and then this should be translated into a protein by the ribosome. So some of these RNA molecules will now contain one of these funny structures in the beginning. That's the so-called 5' untranslated region. And this would then look like this here. And the ribosome that makes the protein requires a certain sequence to start with the translation. And this is shown in red here. But if this sequence is hidden in such a secondary structure, the ribosome cannot produce the protein. So what exists actually in bacteria are riboswitches, where a small molecule ligand can actually bind and to such a structure, and by that actually it will stabilize an alternative secondary structure. So this thing here could actually then assume an alternative structure which is more stable in the presence of the ligand, and this is shown here. But when this form is attained by the molecule, then this red sequence, the RBS, is suddenly accessible, and now the ribosome can bind. It binds here, and then it will translate the rest of this RNA sequence and produce a protein. That would be then counted as the output. So the ligand in this kind of context, it would be called the input, and the output is a protein, which is shown here. The AUG, just for your understanding, that's kind of the so-called start codon. So in translation, what is required is this binding site for the ribosome, that's the RBS, and this little sequence, which is called the start codon. And from the start codon codes for the very first amino acid in the chain of amino acids that will then form into the protein. Now you can actually combine that with this trick that I was just talking about, and this is shown here. So you could now, instead of hiding the ribosome binding site in such a structure, you could place it in a hairpin. And now also the ribosome cannot bind here because it's too big. It cannot really access the RBS. The AUG is here, that's the start codon. And here you would have one of these kind of structures actually similar to the previous slide here. So it's more or less the same as here. Now an RNA molecule could come in, bind with the orange sequence to this A sequence, and then it will actually invade that stem that leads to such a structure. Then the hairpin breaks open, the ribosome binding site is accessible, and the ribosome will produce a protein. Now this is an artificial system that is called a toehold switch, which was also which was developed by several people like Jim Collins, and then later further developed by Peng Yin and Alex Green and co-workers. And this is something is a popular kind of switching mechanism that actually merges ideas from DNA nanotechnology and synthetic biology. So here is another version of that, and this is something that one of my students kind of recently explored a little. And you see, this is actually a mixture of these two approaches. So my student Mark had the idea to, instead of putting the ribosome binding site in a loop like in this Toll switch, he again put it into like a stem loop like in this original ribose switch. So this part here is the same as here, but now you would actually put another sequence in here, which is actually the complementary sequence to this blue sequence here. And now things become confusing, I'm afraid. So this is called the ribosome binding site. The complementary sequence is called the anti-ribosome binding site, anti-RBS. And in the loop, we have an anti-RBS. So what now happens when you add this trigger molecule, the trigger will bind here, it breaks that structure, then this light blue sequence can actually bind to the dark blue sequence, and then this is what you get. And now the ribosome binding site is accessible again, and you get some translation of the protein. Now, importantly, so this is kind of how it looks a little more on the sequence level. So instead of just drawing these images, in the end, we need to really come up with sequences that do that job. And there is a lot of kind of also thermodynamic kind of calculation programs out there that help you designing these systems. And you can then sort of try to balance the energy. So this thing will really switch so that in the end, what you want essentially is that the switch is stable in one conformation. In the absence of input, it must be separated by some energy barrier from the active state. So it's not just switching on without any input, but in the presence of the input, it must be capable of switching, and then the active form must be more stable. And this kind of requires some fiddling around, and it's sometimes not so clear what the design rules in the end are. And actually nowadays people of course try to also employ modern machine learning techniques to understand how to better design such switches. In our case, my student Mark did it by hand, made many different copies of this, and then you can actually put the genes responsible for such RNA molecules, because in the end everything is encoded in DNA. You make the DNA, put it into bacteria in this case, and then you try to figure out whether it works or not. And so he made different design variations where he played around with the stability of that stem, with the size of the loop, and all these factors sort of play a role. And you see that in some cases you then get, like in the best case, a 4 to 5-fold enhancement in the translation activity in this specific experiment. So, but that's okay, but it's not good enough for many of the applications that we had in mind. So this is still kind of a mystery for us, how to really get a good kind of switch working in E. coli, because initially we were interested in actually detecting naturally occurring RNA molecules inside of the cell. And here, one of the big problems is the leak reaction of that these RNA switches are already on without any input. And this is essentially the signal that you see here. When you don't add anything, you already get some production of the protein. And the problem is that things can happen, like, for example, this RNA molecule will be degraded in the cell already a little bit, that destabilizes the structure, and then the ribosome can bind nevertheless, even though because the structure is already destabilized, or the ribosome binds while the RNA molecule is made before you can actually switch or before you can make the fold into a specific structure. So all kinds of things usually go wrong. Nevertheless, you can actually make like even more complicated systems, and this is something like a gate, a logic gate, a NOR gate that implements 2 of such switches in one molecule, and you actually need because it's a NOR gate, you get essentially only an output of protein when there is no input. So you always get an output when you never get an output when either one or the other trigger or both are present. And so that's, I think it's a rather boring gate, of course, but on the other hand, NOR gates are kind of also in principle capable of making all kinds of gates if you could connect them, all kinds of circuits if you could connect them together. So the mechanism here is actually quite involved because it involves both transcriptional and translational switching. And I hope I'm not losing you if I explain it in detail. So, but the point is that actually there is one site on the RNA that terminates transcription, and this requires a small molecule called Rho. So if this molecule is not small molecule, it's kind of a protein which, when it binds, then it terminates kind of transcription. But what you can do is you can actually hide the binding site for this Rho molecule with a ribosome. So when the ribosome would go there and then sit there and prevent transcriptional termination, and this is something that we actually are using kind of in this, that one of these hairpins actually is sort of switching on or off translation. But if we switch sort of off translation, then the ribosome cannot bind and start moving on the RNA. But if the ribosome doesn't move on the RNA, it doesn't hide the binding site for the Rho factor, and then the Rho can bind and terminate transcription. So that's kind of involved, but in the end, these two things together lead to simply the fact that you don't get any protein as long as you have one or both of these triggers present. So because that was not working extremely well, we thought maybe we are going to make some simple biosensor out of that. And here is another example of, where we actually tried to switch a so-called fluorogenic aptamer or a light up aptamer. So there are many RNA structures out there that actually can bind to other molecules. These are called aptamers, and some of them bind to small chromophores like this DFHBI. You see that you have a kind of an RNA structure here, and here's a small molecule bound to that structure. It stacks between such base pairs here, or G quadruplexes in this case, and when it's bound there, then it becomes highly fluorescent. So this aptamer, in a sense, helps this molecule to become very fluorescent. And it turns out that this binding pocket requires many of these guanines. So there are these all these guanines that make a so-called quadruplex structure. So my student Mark had the idea, why don't we now apply the same trick as before, but now we are going to sort of, instead of the ribosome binding site, we will just put some of the sequence that is required to make this fluorescent or fluorogenic aptamer, we will hide them into such a structure. And so this would be another depiction of one of these RNA aptamers, and here are the guanines that are so necessary for making that kind of binding pocket. Now you can actually just put a few of these guanines here into one of these structures here, and then the aptamer is unable to fold into this fluorescent form. And here you see now this has a really extremely good kind of performance in the sense of fluorescence activation. So here we get an increase of sometimes several hundredfold, simply because in the absence of a trigger, there's no fluorescence at all. So the background is simply the sort of the autofluorescence or background of the medium. And so this is kind of an interesting sensing application. Unfortunately, this does not work in cells so far. So in cells, it works much worse, but maybe this can be done, can be used in for RNA sensing, for example. I see that I'm really talking too much. I need to, I only have 2 minutes left, right? I wanted to
No
just
worries. I think you can just go on. So, so we won't
very
cut you in 2
quickly.
minutes.
So of course, many people are excited about CRISPR recently, and CRISPR also contains RNA, and therefore the same things that I was just talking about, we can also do actually with CRISPR. And for the people who are not familiar with CRISPR, CRISPR originally is actually some type of immune system for bacteria. So the bacteria try to cope with infections by phages or bacteriophages. And what they do, and I'm not going into the detail here, but they actually have a mechanism where they actually cut the genome of phages into small pieces, and they can store them in a so-called CRISPR array. And later on, there are kind of other enzymes that then actually can translate this CRISPR array. This will make, transcribe the CRISPR array. It will make an RNA molecule. This RNA molecule can be cut into small pieces. And then these RNA molecules are bound to a complex, a CRISPR-associated nuclease, typically. And now this complex can actually bind to DNA that contains the corresponding sequence that was originally stored in that array. But that means that actually, if a phage infects the bacteria that already have kind of this, the information stored in the array, then this mechanism will recognize the genome of these phages, and this CRISPR complex binds, and it just cuts the genome, and therefore essentially avoids infection by that phage. Now, this is a really great technology because you can, in principle, direct this protein complex to a specific binding site on a DNA molecule by simply choosing the correct RNA molecule. And these RNA molecules are called the guide RNAs. And there are many potential applications of that. So you only need to design these guide RNAs. They bind to these CRISPR proteins. They bind to your target sequence, and then they can either cut the sequence at a certain position, or you can just place them there without cutting, and then they just form roadblocks that then actually influence, for example, transcription processes. So what we have been working with in the lab for the last years is a version of CRISPR that's called CRISPR-Cas12a. So most of you probably have heard of Cas9. But Cas12a is an alternative, and it has the advantage that it has a rather small guide RNA. That's shown here. That's only a short RNA molecule. And the sequence in that molecule here determines where your CRISPR will bind on a piece of double-stranded DNA. So this is indicated here. So what we did is we now actually combined this trick with toehold-mediated strand displacement with this CRISPR mechanism. So this molecule over here, the red sequence, is called the, sorry, this sequence up here, that's called the handle. And it's called the handle because the CRISPR protein binds to this so-called handle. And the handle is what we show in blue here. That's the handle. And we can actually make a structure that looks like this. And here, the handle is actually prevented from forming by just hybridizing it to a complementary sequence. So in this form, this RNA molecule cannot be recognized by the CRISPR protein, and nothing will happen. But what we can now do, then, that's the same trick as before. We add an RNA molecule. This kind of yellow sequence will bind here, it will break that secondary structure, and that leads to this double strand. And then actually the rest of this, the H sequence can now form this handle sequence. And now this sequence is actually recognized by the protein, it binds here. And then this has a funny property that it actually cuts at the position of the red star. And you end up with such a complex And now this complex will bind to the target DNA and cut it. And that this works is actually shown here. So we have a target DNA that is recognized by that sequence. And this is showing a gel electrophoresis image. So usually we separate DNA molecules by the size, by running them in a gel in an electric field. And here you see a very big molecule. And if you cut that molecule, you get smaller pieces that are shown here. And you see that we can, by adding this trigger molecule, we sort of induce that transformation. Then the CRISPR binds to its target and cuts. And then we see in the presence of trigger, we actually have a smaller molecule, which indicates cutting. So that's actually great. And we can actually make more and more complicated systems with that. And just as a example here, now we can again try to do some computation. Now we actually can construct or make a system where we first need to bring several molecules together to actually induce this conformational change. So what happens here is that there's a trigger part number 1 and number 2, and when they both are present, then this AND part can bind together. Then you have this form. But only when you have that complete molecule, this toward can bind here, and then the rest of the sequence, this split handle sequence, can actually invade here and break that structure. And then you get this structure. And now, again, the handle can form, the CRISPR protein can bind, it can cut here and then activate sort of the next step. And this is shown in this gel. So again, we add the 2 triggers. When we don't add them, nothing happens. When we add one of them, nothing happens. When we add both of them, we indeed get activation of that CRISPR system, and that's a very kind of clean AND gate. It's actually much better than the other system that I showed you before. This is why I guess also many people are excited about CRISPR in general. I'm really running out of time. I just wanted to mention that similar things can be done in mammalian cells, and then things become more complicated. But so here's one example. In this case, okay, sorry that I'm jumping back if I try to explain how that works. So in a eukaryotic cell, you actually need to you have a cell nucleus, and some of the RNA molecules are actually exported out of the nucleus into the cytosol out in the exterior. And because there the protein production takes place. And in order to be exported from the cell nucleus, you need something called a cap. So this is made by the proteins in the nucleus, the 5' cap and the poly A tail. So when you have that, then your RNA molecule will be transported out of the nucleus. Now we had the idea to maybe cut away. So first let these RNA molecules be exported from the cell, and then we cut this tail off, and when the tail is missing, then actually the RNA will be kind of localized in the nucleus again. The details are a little more complicated, but that's the general idea. So what you can do, for example, you can make systems like this, where the CRISPR protein guide RNA is kind of exported from the cell and the nucleus, and then another kind of complex called RISC that comes from RNA interference will actually cut away this piece, and then the rest of the molecule will again be bound by Cas12a, and this will exert some function in the cell nucleus. So you might wonder why this is interesting. The point is that these little molecules, these yellow molecules, these so-called microRNAs, they are present in different cell types at different levels, sometimes in certain cancer types at different levels, or in certain tissue types. And therefore, in principle, with such an approach, you can make the action of the CRISPR system dependent on the cell type, or you could kind of program systems where you say only when it's a cancer cell, I will kind of perform some other function. So this is at least in principle possible. And this is what many people are dreaming of, to make sort of conditional kind of CRISPR systems in order to make them more specific and safer, and so on. And as I'm running out of time, I'm sorry. I realize that with increasing age, I tend to talk longer and longer. I need less slides per talk. But so maybe just to end, how all of these things would actually come together in a molecular communication context. So we are really focusing on nucleic acids because they are kind of programmable. It's kind of easy to deal with sequences. We can calculate their properties up to a certain degree, calculate these structures, thermodynamic properties, but they are not maybe the best for communication between cells or communication between cells and maybe synthetic systems. So this is something where you can probably rationally design certain computational functions inside of the cell. But of course, at one point, you need to get this information out. And this could be, in the simplest case, it's some type of fluorescence or luminescence signal, but maybe you also want to communicate with other cells. And there are essentially different options available. So one thing is that you could couple such a genetic circuit to, for example, the expression of a cell surface protein that will then interact with another cell surface receptor of another cell. So that would involve direct cell-cell contact. But here you could, in principle, use one of these kind of RNA-based kind of switching events to directly induce the communication with a neighboring cell. But maybe other mechanisms would be that, for example, you could generate, like you will produce an enzyme that will generate a small molecule, or and this small molecule could diffuse from one cell to the other, where it is actually received by some transcription factor that takes that as an additional input, and then you could activate gene expression in a cell very far away. So this is another way how this could be connected. And there's actually also a funny mechanism that is also used in nature, where actually RNA is packaged into small vesicles called extracellular vesicles or exosomes that apparently also are used in biology for communication between cells. They can actually communicate via microRNAs, or at least that's kind of thought to be what's going on. So you could imagine like packaging microRNAs and sending them to other cells where they then exert some regulatory function. So I guess therefore, whatever I've told you today, so trying to make like RNA-based switching mechanisms and maybe small computational elements and circuits will be only a part of a bigger kind of course, system where you need all kinds of other kind of components to really establish communication between cells or between cells and synthetic systems. And with that, I'm at the end of my talk, and thank you very much for your attention. Thanks.